My name is Anton Zavyalov. I am the director of Joint Biotechnology Laboratory at the University of Turku, Finland. The title of our poster is Secreted Bacterial Adenosine Deaminase is an evolutionary precursor of adenosine deaminase growth factor. Adenosine has many important functions inside of living cells. And at the same time, it acts as an important signaling molecule. The concentration, the level of adenosine must be tightly regulated both inside and outside of cells. This is achieved through the activity of enzymes called adenosine deaminases. There are two distinct adenosine deaminases. One is a cytoplasmic protein, ADA1, and the other is a secreted protein, adenosine deaminase growth factor, ADA2. Cytoplasmic ADA1 plays a key role in purine metabolism, and it's widely distributed from prokaryotes to mammals. On the other hand, secreted ADA2 is an important cell signaling protein that was thought to be present only in multicellular organisms. The aim of our study was to reveal how secreted ADA2 has emerged in this advanced species. Previously, we showed that ADA1 and ADA2 are structurally very different enzymes. Whereas ADA1 is a relatively compact monomer, ADA2 is a homodimer of subunits which, in addition to the catalytic domain, have dimerization and putative receptor binding domains. In addition, ADA2 has a signal peptide, and it's heavily glycosylated and stabilized by disulfide bonds. This large structural difference between ADA1 and ADA2 makes it highly unlikely that secreted ADA2 evolved from eukaryotic ADA1. In hope for finding bacterial homologue of ADA2, we performed a basic BLAST search of the NCBI protein sequence database against human ADA2. This, surprisingly, this rather basic search retrieved sequences of a family of bacterial proteins, all from the recently sequenced genomes of gram-negative bacteria from the phylum Bacteroidetes. Using a PCR analysis of selected species, we confirmed the presence of this protein. These bacterial proteins form a well-defined branch on the phylogenetic tree of adenosine deaminases. And it's very obvious that they are closely related to eukaryotic ADA2. The focused phylogenetic analysis of ADA2 proteins revealed that bacterial ADA2 are close evolutionary close homologs of ADA2 from lowest eukaryotic species. Bacterial and human ADA2 can be nicely aligned without significant gaps, and these proteins share about 30-32 identical and 68 homologous residues. This is rather significant sequence similarity. A homology modeling based on our crystal structure of human ADA2 predicted that bacterial enzyme also has the dimerization and the putative receptor binding domains. Also, this modeling suggested that bacterial and eukaryotic ADA2 share a very silica catalytic site. Thus, our in silica analysis suggested that bacterial and eukaryotic ADA2 are close relatives, and it predicts that these proteins have very similar catalytic activity. They form homodimers, and they are secreted to the extracellular space. Also, it's possible that they bind to the same biological surfaces via this putative receptor binding domain. To verify our prediction, we biochemically characterized ADA2 from Elizabeth Kingia Anopheles. We cloned the gene in Escherichia coli. It efficiently secreted to the periplasmic space, and the purified protein showed strong adenosine deaminase activity and had catalytic parameters very similar to those of invertebrate ADA2. Next, we studied the native endogenous enzyme from E. Anopheles. This bacterium has no the ADA1 gene in the genome, but we detected strong catalytic activity both in the lysate and culture medium of this bacteria. We also were able to detect the native enzyme using antibodies raised against the recombinant protein. The extracellular native protein was fully active, and it has catalytic parameters identical to those of the recombinant protein. It also, we also studied the gel filtration of this protein, and it migrated on gel filtration columns with molecular weight consistent with dimer. So we can conclude that we discovered a bacterial homologue of ADA2. Bacterial and eukaryotic ADA2 possess the dimerization and PRB domains characteristic for the family. They have nearly identical catalytic sites and show similar catalytic characteristics. Most surprisingly, the bacterial ADA2 has a signal sequence similar to that of eukaryotic ADA2 and is specifically secreted into the extracellular space, where it may potentially control the level of extracellular adenosine. And this suggests a potential role of ADA2 in bacterial communication. Our study has already been published, and you can find our article in Journal Molecular Biology and Evolution. Thank you for your attention.